also one. We have also one in French Polynesia, and then we have a learning site covering the whole Europe in order to compare and to try to harmonize results across the different regional seas and across the different learning sites. Please go ahead. For the data availability and management, etc., we have the data management plan updated recently. You can go to this link and to download. It's deliverable 5.5, and you can see in the figure the data flows. And also we have created a Zenodo repository in which we are providing all the information, all the data sets we are creating, as well as in the supplementary material of the papers we are publishing. Until now, we have published 50 papers. Some of them have supplementary material with data. And in this case, we are keen to collaborate with EMODnet in case we can provide this. Other information, for example, we have published a data set, a template with near 2,000 microbial taxa and with ecological groups to assess the status, which is publicly available. So if EMODnet is interested, we can provide also this. We have created the list of near 2,000 taxa with ecological groups for the benthic communities. We have provided this to worms, but it is available also in that you can see there. This is to calculate AMBI, which is the AZTI's Marine Biotic Index. And finally, we have recently submitted the paper you can see at the bottom on a data set from 30 years of information coming from the Basque Monitoring Network, including water, sediment, biota, phytoplankton, macroalgae, macroinvertebrates, and fish from estuaries and coasts. And we are going to publish this, I hope so, in the coming months. And of course, this information could be also uploaded into EMODnet if you are interested. And that's all from my side. Thank you.
Thank you very much, Angel. Johanna, do you have any questions? Because I think that those data could be very useful to integrate the information. So then maybe we should be in touch later on.
Okay.
Sorry, I'm moving rooms.
Okay.
Yeah, actually, I was just telling my colleagues here, Ruben and Lynn, that yeah, I think it might be interesting. However, what we have to make quite clear is that we don't have a connection to Zenodo. So the data are stuck there. What I would encourage, if possible, Angel, is that and any others that use Zenodo or other repositories for your data, that if possible, you reformat it to Darwin Core. Because if we eventually manage to get a connection to that repository or others, and if the data are already in Darwin Core, self-described, and with all the metadata that is needed for it, then the process is slightly easier to incorporate and to publish in EMODnet Biology. So, because Zenodo just accept the files instead of just try and make it compliant with Darwin Core. That would already be quite a step forward. But yeah, we don't have a connection to Zenodo. It's so yeah.
Okay. In any case, I will contact my my data managers at the project, and we can be in contact.
Yeah, I guess so that you don't have to do the work twice. If you put the data in Zenodo in Darwin Core, and then you point us to those data, or you create the IPT resource for those data, you can reuse those same files. So yeah, so that's a way forward that can be discussed when you're ready to do that. But,
okay, good. Yeah. Thank you.
No problem.
Maybe I can make a little addition to that one. So if you're dealing with GBIF data and Darwin Core at the European and Central Asian level, we also have 2 support officers, which are positioned there to help data publishers formatting their data in Darwin Core. And probably Ruben at EMODnet can help you as well, and Bliss. But there's support there to format your data to Darwin Core.
Yeah, the problem, Niels, is that we don't have resource to reformat data for these projects.
Okay, but at the level of Europe, GBIF does have. There's 2 data support officers which can help. Yeah.
Okay. Thank you.
Thank you. Thank you. So we can proceed to biodiversity meets data, please.
Yes, thank you very much. So Biodiversity Meets Data is a new project. We'll only start in March next year. So I'm Niels Raes. I'm also the project coordinator. The aim of Biodiversity Meets Data is to develop a single access point to biodiversity data and analysis tools for managers of natural resources and policymakers across Europe. So that's answering to the Digital for Nature call of last year. And we'll collect eDNA data, but also high throughput biodiversity monitoring data from camera traps and audio devices. And we cover Europe and to some extent the global level. Next one. So it's a single access point. It's answering to the needs to report under the nature directives of the European Union. So the Habitats Directive, Bird Directive, Freshwater Framework Directive, and Marine Strategy Framework Directive. On the right side, you see the full work package structure. And on top, which is the most important one for all the end users, is the single access point. Next. So we'll provide high throughput biodiversity monitoring through image and sound capture, eDNA sampling, and this is combined with AI taxon identifications. So at the right bottom, you see the high throughput data generation. Next one. So, and then this data is being generated. This includes all kind of data. And then we also mobilize legacy data from historical baseline resources. We use GBIF and Darwin Core as the data repository. And in that sense, we can directly link with EMODnet and OBIS. So we also have a data catalog that's on the lower left. And hopefully we can link there to some of the spatial data layers that are provided by EMODnet, which are relevant for the marine domain. So we're dealing with virtual research environments. So that's combining this biodiversity data with all the environmental data, which are harmonized in space, time, and taxonomy. These cover 3 domains, and for EMODnet, the marine domain is probably the most relevant, and that's where we hopefully can link with the end users. So these VREs facilitate biodiversity monitoring, trend analysis, the analysis of drivers of change, and the projections of land cover and climate change for these areas. The final results will be made available, visualized in a map viewer. And the workflows will be documented through Workflow Hub, which is basically developing fair workflows. So you can reproduce the entire workflow with the VREs. Next. So, and very important in this sense is the stakeholder and user engagement and the mapping in the mapping of all the stages of the single access point. So we need interaction with users of EMODnet, what kind of analysis they would like to see from the VREs, how they would like to approach the single access point, and this is in full co-design. So the left side you see work package 1 is the full co-design. On the right side there's the training and communication and dissemination. So we'll organize a lot of training workshops with this project. And then the last one. So and that's will start next year in March. That's what I can share for now. Thank you.
Thank you very much, Niels. And I have a question. Yes. Is this a project or a longer-term initiative? I mean, how will this single access point be maintained?
Yes. So this is a 4-year project. We guarantee maintenance an additional 5 years. Well, we are in close contact with the EEA and GSC. So hopefully we can transfer some of these services to our next level after the project has been finished. For the data, we're looking into connections with EOSC and the Green Deal data space to continue work there, and well, possibly we'll collaborate, and well, likely we'll collaborate with EBOC as well on this front to see where we can harmonize things in the course of the development of this project. But we aim for linking with many of the biodiversity initiatives across Europe, so we are very open for collaboration, and all the work we do is in open data and open source format, so we are happy to share anything that we develop. Yes.
Thank you very much. Are there any comments or questions, or we move to the next presentation? Yes, Protect Baltic, please.
Hello, my name is Kimmo Koivumaki. I'm the data manager in this Protect Baltic project. I'm representing also HELCOM, which is the project coordinator. Our project consists of 7 countries which are surrounding the Baltic Sea. Russia is not the partner because it's an EU-funded project and because of the geopolitics at the moment. Poland is not the direct partner, but they are closely also following up us through different HELCOM groups and then this project advisory board. And what we are trying to do is to build this kind of ecologically reasoned and effectively managed MPAs in the Baltic Sea. MPAs being these marine protected areas. And this presentation now is about this project data call, where the data was requested from these project countries and collected from multiple different open sources such as EMODnet. Then the data is utilized by these partners of the project in their work and publications. And some of the data will be published in our own project developed portal, MPA portal. And then there is also other type of data requested in this project, such as data straight from these different marine protected areas. But this presentation is about this project data. Can you change the slide? Okay, so we originally asked about 14 different data sets. And a large part of the data was something that has not been requested before. So the availability from different countries was unknown. And for this reason, it has caused also additional work, and it has taken more time than the normal data calls, what we are doing at HELCOM. And this data call is still ongoing, but we are trying to finish it this year. And then the majority of these spatial data collected by the project will be stored in our HELCOM's map and data service portal and also biodiversity database. And this biodiversity database is basically species observations data, and it is Darwin core compatible. And of course, we are willing to then share it for you as well, if needed after the project. Majority of our data is open using this Creative Commons license. And then when we have some restricted data from these project countries, it can be used for modeling and analysis, because then the data details are kind of they are not anymore visible there. And all these cases where we are using partly even restricted data, then it will be handled specifically case by case by these partners. Some of the data have their special disclaimers, but generally it is open. Then the data is harmonized when we get it, have been collected it, and then the metadata is created by us in HELCOM if it has not been provided by these different sources. Then the project experts will use it, or then we will publish it in the portal as such. Metadata is stored in our metadata catalog, which is similar to yours, EMODnet's. And then from EMODnet straight, what I could find was this sedimentation accumulation rate, sediment maps, and then total suspended matter, which was used as a proxy for our turbidity dataset. This sedimentation accumulation rate had data from some countries only, and then the sediment maps from EMODnet Geology got seabed substrate, and it was generalized to one in relation to 250K resolution, and then we clipped it for our sea area, and then the turbidity was from EMODnet ERDAP, EMODnet Physics, and that was a huge file which we didn't standardize in our end at all, but the expert says that they are able to download it straight and use it for their purposes for the moment. But that is now our current relation with EMODnet. Thank you.
Very much. Thank you very much. So, as you say, the data are restricted, so they cannot be available, or is it? Did I understand?
Yes. Some of the data, like depth data, for example, it is not freely available necessarily from all countries, but it can be used when we are doing models and when we are doing analysis. So then in the results, it's not anymore visible like the actual depth, but it can be used as a proxy during the work for the experts. And sometimes if it's not available, it's always discussed case by case that what can be done with the data. And if it's not suitable for our purposes, then we are not using it.
Okay, thank you. Please,
I have a related question to the restriction. So because we're facing similar difficulties with restricted data and we're looking for solutions. So, but how would you treat this data after the project has been finished? So where do you maintain this restricted data, and how do you prevent that the data gets lost?
Yeah. We have our own internal server where we are having this original data, so that one we don't give even to the experts. So for the experts, we are delivering only the openly available data, and then this standardized data where we have already done our verification rounds. But the original data will be stored in HELCOM's internal server, and they have been there for dozens of years already, so it is basically there as long as HELCOM is alive, at least. That's all I can say, but...
Okay, thank you for that. Yes.
Yep.
Thank you. Any comments? No, so we can proceed to the last project for today. So DG for Eco.
Hi, hello. Can you hear me?
Hello, yes, we can hear you. Hello. Okay.
So I'm Clavel-Henry from GEOMAR, and I'm going to pitch on the Digital Eco project funded by the EU OBIS-env program and coordinated by Koivumaki in the ICM Spain. Next slide. The Digital Eco project is working for the development of digital twin of oceans, which is a way to simulate the marine ecosystem, to understand and to represent the past, present, and future states of marine biological and environmental components to improve marine management strategies. So before arriving to those strategies and before to arrive to reach the functional details, we need to collect data and data that are enough representative of the marine ecosystem. So for that, we need to define strategic areas where to repeat measurements and monitoring activities. So Digital Eco is going to implement robotic networks at strategic areas to get real-time data, and as well to collect historical and sweeping data being the one that you keep after the surveys, the one that came from fisheries and all that. And as well, with those data, we are targeting for the upscaling along the temporal and spatial dimensions. Next slide. Again. Yes, thanks. So we are going to develop deployed platforms that are multi-parametric sensor to collect various and diverse data. We can classify those platforms by being fixed, static, and getting data along the temporal dimension. And we can have platform as well classified as geographical or mobile, collecting data along the spatial dimension. Next slide. Yes, thanks. We have 4 strategic areas in European water, coastal waters, that are going to use similar platforms, but that are going to collect different biological data based on the ecosystems that we have in those areas. So in the Irish and Swedish coastal waters, we are targeting to look and obtain data of presence for novel lobsters and seals. And for the Mediterranean strategic areas, we are planning to collect and detect data for parrots, fish, and dolphins. Next slide. So the Digital Eco project has a data flow that is presented there from the collections and acquisition of data to sharing the data to the customers. Next. Yes. Thanks. So for the data, we are going to collect biological data through imaging, eDNA, passive acoustic monitoring, and tracking animal wearing some tags, acoustic tags. And with both information, we are going to have as well environmental data, as you know, weather, oceanographic, and geochemical. And with other biological data that we are going to obtain are the sleeping data, and for that we have calls to stakeholders to share that with us. Then once we have the data, there is a role data management plan well defined and required by the European programs funding, including as well the way to filter data, harmonize, format, check for the quality control, and standardizing. And then all those data will be merged for the user to access in the GUI, and as well connected to EMODnet in a certain way. And that's why I'm here as well today, to glance and to catch information. Thank you for your attention. That's it for me. No, the rest is just like a summary.
Okay, thank you. Thank you very much, Moran. So are there any feedbacks from Joanna? No? So we can go to the next part of our meeting, which is the presentation by Ruben and Lynn on EMODnet Biology data resources. So I leave the floor now to you, Ruben. Thank you.
All right, so can you all hear me? All right. I'm gonna change some settings here, and you all see the screen, right?
Yes.
Well,
yes.
That's right. So we can start. Yesterday, as you remember, for the ones that were here, we were talking about the FAIR data principles and research data management. Today, we have a similarly long presentation on the guidelines for the data that need to be submitted to EMODnet Biology. So we will talk a little bit about how you should format your data sets or work with your biodiversity occurrences data sets if you want to send them to EMODnet Biology, and what tools you can use to do that. I'm not presenting alone today. As you can see, here is my colleague Lynn Delgat, also from the Flanders Marine Institute. And for the ones that were not here yesterday, I'm Ruben Perez, also from VLIS. She will be presenting on the eDNA data that we will see. So we will have a section for that since it's being requested by a number of you. So let's delve into it. All right. So I have, like yesterday, a number of sections for this presentation. At the beginning, I will provide a little bit of background talking about the data flows of EMODnet Biology, where does data come from, and we will talk about the data standards, so Darwin Core. Later on, we will go with Lynn through the DNA-derived data submission to EMODnet Biology. And afterwards, we have a couple of small sections that are a little bit more dynamic because they have demos. And they are about publishing data, not only in EMODnet Biology, but also in GBIF and OBIS, since we will be using the IPT tool, and also the quality control that we do in EMODnet Biology, also using a number of available resources. So let's go. Oh. Maybe Marina, if you can mute everyone.
Sistemde vekil kullanıcı diye bir şey var o izin formunla.
Thank you. And apologies about that. Let's continue about data flows. I'd like to provide a little bit of background, complementing what Joanna already mentioned yesterday. EMODnet Biology started in 2009 and working as a wrapper around the European node of OBIS that would use the EUROBIS infrastructure as a backbone, and it would develop from it a number of services, right? Since then, EMODnet Biology, it's been powering the European node of OBIS with occurrences data from all type of sources. So it can come from traditional data sampling surveys, from monitoring programs. It can come from citizen science. It can come from different technologies like acoustic telemetry, satellite tracking, and recently also eDNA. And currently, we have more than 1,400 data sets, more than 40 million occurrence records, and more than 100 million measurements and facts associated to those occurrences and events. And we will explain what those events, occurrences, and measurements and facts are a little bit later. But this helps. This is some help that the EMODnet Biology has provided to EUROBIS to become one of the most active nodes of the whole OBIS network. Now, while EUROBIS had a connection already with OBIS and therefore with GBIF, EMODnet Biology has brought a network of organizations to power and the power to develop all this data flow, right? So from EUROBIS, from the data that comes from this consortium of EMODnet Biology goes to EUROBIS, and from there it goes to OBIS and the GBIF networks. Now, this data do not only come from these sources, from the consortium members, but they also can come from many other places or repositories. For example, other lots of EMODnet, like EMODnet Cyber Habitats, or the data ingestion portal of EMODnet, also from independent data providers that do not work with any projects, but they want to see their data sets also be made public. But also we gather data from other OBIS nodes or from GBIF itself, right? And we integrate it into EUROBIS and therefore into EMODnet Biology. Now, eventually, everything makes it within the whole EMODnet network to what it's known as a central portal, which combines all the thematic lots of EMODnet, allowing for more specific and complex queries on the data. So not only biological, but it allows to also gather physical information, chemical information, etc. In this way, EMODnet Biology makes a data flow from data creators to data users. Now, this data flow, and this is something that we commented slightly yesterday, it's only possible, this integration, thanks to the use of common standards. And here we adhere to the widely known and community-used Darwin Core standard developed by Tedwig. And at the beginning, we started in EMODnet Biology focusing on documenting only occurrences, presence only, presence absence, and a few parameters. But with time and a lot of community effort, we reached the current format that we use, which is known as the OBIS-env data format. We will delve into it a little bit later, which allows to document not only taxonomy, spatiotemporal coverage, and also other parameters that were already there, like biomass abundances and life stages, but it also allows us to document in a standardized and an interoperable and efficient way information about sampling methodology, sampling effort, environmental data, such as temperature, salinities, together in that one dataset. Now, for this, we do not use only the Darwin Core standard, but we also use a number of other control vocabularies, such as the WORMS, the World Register of Marine Species for taxonomy, as we said yesterday, marine regions for geography, or the BODC NERC vocabulary server collections for all these biotic, abiotic, and metadata parameters that are associated to those events and occurrences, and contributing to make the data a little bit more fair in the interoperable aspect of it. Now, let's go into Darwin Core, into the data standard that we use. So Darwin Core, for the ones that are not yet familiar with it, it works as a data standard. It's a set of guidelines intended to facilitate the data sharing of information about biological diversity. It's based on the occurrence of data in nature and that is documented by either observations, collections, or detections. Now, here, Darwin Core, what they do, they set a specific format on how a data set should look like, should be formatted. And they're going to specify a controlled vocabulary list of names that we will use as the field names of our data sets, as the column headers. But they also are going to define the file structure that a biodiversity occurrence data set should have. And this is how we reach the first part of Darwin Core, which is the Darwin Core term, that list of controlled vocabularies that will be used as the field names, as I said before. They're all written in a very specific format, so lower camel case. They are well maintained, they're standardized, they have definitions and some sort of other attributes. And you can see here in the example a number of them. And in one of them, for example, basis of record, we would use this field in our table to specify the nature of the occurrence. For example, if that occurrence has been extracted from a machine observation, if it comes from a collected specimen in the field, or if it's a fossil, for example, in a museum. This will help us to distinguish this information. Now, together with the list of terms that Darwin Core provides to become those field names for our datasets, Darwin Core also provides a method for the structure of our dataset itself. Darwin Core has proposed the use of what they call Darwin Core archives. A Darwin Core archive is a data package, is a zip file that contains all the relevant information about datasets, including the metadata. So it contains a number of data tables. It's going to contain a meta XML file, and it's going to contain an EML XML file. We will go into them a little bit later in this presentation. So traditionally, the data tables of Darwin Core datasets will follow what it's known as the star schema, where the dataset is represented as a variable, as a variable number of data tables. So it's not only one, it can be many of them, and that's going to depend on the datasets you have. One of these tables is going to represent the role of a core data table, and the other ones will be extensions that link to it, link to the core and provide extra information. Now, this aims to allow for a lot of flexibility, as you can imagine, since depending on the data set, we can organize these tables in a different way, but it also helps to keep the structure of our data packages of these data sets simple enough. However, of course, it has limitations, just like all the other methods, such as high levels of redundancy. The good news is that this has been partially solved by the specific format that is called the OBIS-env data format, that connects the extensions to each other, so it breaks the star schema to create a more complex relational database. This is an example of an OBIS-env data format dataset. Follows Darwin Core structure, but as I said before, it connects the extension tables to each other, so it doesn't follow anymore the star schema. In this case, in this example table, the dataset depicts a water sample, the west taking, where species presences were extracted from there, but they also measure temperature data. So in this table, for example, you have three different tables. One of them we'll call the event table, the other one will be occurrence table, and the EMOF, or extended measurements or fact tables. These will be one of the structures that your dataset can have, right? In the event table, we will have information such as the event date, and of course an identifier. to connect to the other tables. In the occurrence table, we will have information about the taxonomy, the identifications, and some more information about the organisms that have been collected. And in the EMODnet table, we will have all those parameters that have been measured together with the occurrences and the events, such as temperatures, lengths of the animals, etc. Now, the meta XML file, which is also contained in a Darwin core archive, it's like a README file. It shows us the relations between those data tables, right? It's going to show us also the format of those data tables, and it's going to provide links to the Darwin core list of controlled vocabularies that are used as column headers in our data tables. This is going to make the data set better understood, interpretable, and well documented. And this all comes within the same zip file. So everything is contained. The last file that the Darwin core archive has is called EML file. It follows the ecological metadata language format standard. And it has all the metadata of the data sets. So it's going to contain the title, the citation, the license, the description, all the contributors, etc. Now, you do not need to create these XML files yourself, because we have tools, GBIF has developed tools that we will discuss later to develop all these tables. Same thing goes for the meta XML file. So you don't have to deal with it. Although if you want, you can create your own Darwin core archive files. Now, there are many ways in which a dataset can be structured in Darwin core because it's very flexible, and that's going to depend on the type of data that it contains. For example, if you have parameters that are linked to the events, such as environmental information or sampling information, we'll have a specific given number of tables. If your dataset contains stomach content or parasite host occurrences, it will have a different structure with different tables. If it has fossil biodiversity, for example, it will contain other tables. And choosing new tables, again, will depend on the datasets that you have. Luckily, OBIS has designed a decision tree that helps us to choose which tables we should use. So, for example, if you have here biomass or abundance, we'll go in this line, and then if that's what our dataset contains, but it only contains that information, we would go for an occurrence core with maybe an EMODnet measurements or facts extension if we have biometrics data, for example, about those organisms that have been collected. However, if environmental information or sampling related information has been collected, we would go back up here and we will use an event core with an occurrence extension or an EMODnet extension. So as you can see, there are many different ways in which the tables can be organized. These are the tables that can be used, or we are using as core tables. And on the right side, you see what tables can be potential extensions. But again, the number of these tables is going to vary a lot, and it's going to depend on your data sets. Although there are generic guidelines on how datasets usually look like. And for that, we can have a conversation later and see what's better for in your specific cases. Now, as I said before, a special type of the Darwin core dataset formatting is the OBIS-env data format, which, as I said before, it breaks the star schema, connects the extension tables. Now, the advantage of it is that it allows for less redundant datasets that have event-related data, such as sampling methodologies, environmental data, while it can still hold biotic parameters about those occurrences in an efficient and interpretable manner. We do not have a lot of time today to go into detail, although I'll have a small example later on how we can turn our datasets into this one. Now, talking about the OBIS-env data format, which is the one that we use in EMODnet Biology, the event table is going to contain one row per event and as many events as necessary. Now, what is an event? An event, it's an action that occurs in a particular place and time. And as you can see, this definition is very broad. Now, events can also hold other events within them. As you can see in this example, we're going to have a cruise, which could be an event where we are going to document the ship name, for example. And in there, we're going to have, for example, station visits. And in each station, we can have several deployments or measurements or samples, right? So each of those measurements, samples, transects, station visits, and cruises is going to be an event, and we are going to use event hierarchy within this event table to relate some to each other. Now, this can be a little bit complex, but it allows us to reduce the size of our data sets by a lot. So it helps a lot when we're dealing with very large data sets or when we are aggregating data sets into big data systems like we do in EMODnet Biology. The next table is the occurrence table. And just like the event table, this one contains one row per occurrences. Now, occurrences are aggregation of organisms, right? An occurrence can be defined as the existence of an organism or a homogeneous group of organisms at a particular place and time. Again, a very broad definition. Now, the number of occurrences here for you in your datasets will depend on the ability of the researcher to create or to make those homogeneous groups. For example, if you are collecting copepods, but you also know that you have 20 copepods, but you also know that 3 of them are female and that 2 of them are juveniles, then you would have one row in that table, one occurrence per combination of sex, life stage, and taxon, right? So it's also very flexible, but it allows to document our data sets with high precision. Now, the last table that I'm going to present within the OBIS-env data format is the extended measurements or facts table, or EMOF. And this one contains one row per measurement or fact. Now, what measurements or facts are, you can see them there in the images, either organism quantifications, they can be abiotic measurements, they can be species biometrics, sampling methodologies, organism characteristics, or habitat description. So it also offers us a very varied way of documenting the data. This table is going to be a little bit special because it's a key value pair table, so it's expected to be very long, and it's linked to the occurrence and the event tables through an event ID column and an occurrence ID column, right? Something interesting about this is that we're also going to have a number of columns here that will contain controlled vocabulary. These controlled vocabularies will come from the BODC terms, and they will standardize the measurement types, for example, life stage, but also the measurement values, for example, juvenile or adult, and the units as well, if they exist. So it provides an extra level of interoperability than we had before in Darwin Core. So it can be very, very beneficial for a number of you if you have these type of data sets. I would like to continue with a very short example of how we can transform a data set from a raw version to a Darwin Core, actually to an OBIS-env data format, Darwin Core style. As you can see, in this table, we have spec name, whatever that is, a date, station, depth, gear, WWB, and life stage. This is the type of data sets that we receive, and sometimes a lot more complicated. This is a mock example that I created. And as you can see, a number of information here is not very clear. So this species is M. asterias, could be anything M. canis. So we would have to go back to the researcher or the originator to know what it is. Some of the information here, like the depth, contains 2 different formats, so either a hyphen or a 2 in character to depict the range, the range of the depth. So we would also need to standardize that. We would definitely need to change this this field name to make it more explicit for the people to understand. And also we would need to remove the abbreviations here. These are a few of the first issues that I found with this one. How we could document it. Let's see it. First step, it's to add the Darwin Core fields. So we can set another row here with the equivalent Darwin Core terms from the control vocabularies of Darwin Core to each of our fields. So for example, spec name will be transformed to scientific name, date will be transformed to event date, and so on. But we're also going to add to our data set a number of mandatory fields. Now, I have here a list of mandatory fields for EMODnet Biology, and this will vary depending on where you're sending your data to, but these are a very basic set of fields to make your data set a lot more understandable. Once we have that, the next step is to standardize some of the fields. So the event date here, for example, is going to be standardized to specific standard that we've chosen in Darwin Core, which is the ISO 8601. And the life stages will also be standardized. So here we will remove those abbreviations and we will add the full words. The depth ranges will be divided into the minimum and the maximum, so which will help other tools to create filters that allow us to filter easily through depth, for example, if we would want that. And we will also enhance our dataset. And here we will add information such as the dataset name, the institution code, and also information that maybe was not there, such as an event ID for our events and an occurrence ID for what to represent our occurrences. And other information such as the occurrence status that tell us if a specific occurrence is documented as absent or present, and the basis of record, which I explained before. After we have standardized and enhanced this dataset, we're going to keep on doing some more standardization. However, I separated this slide because talking about taxonomy and geography, there are a few more things that we can do. And the good news is that we have a number of tools here, like worms and marine regions, that can help us to bring more information into our data set. So after a talk with the researcher, we realized that M. asterias was actually Mustelus, so now we know. But we've also added the scientific name ID, which is the worms identifier for our species. And we've also added the coordinates and the coordinate uncertainty in meters, which we extracted from the locality. And this is one of the functions that Marine Regions offers. You can give them a locality and they give you the coordinates of that place, if it is in their database, of course. Now, we're almost in the end. The last step that we would need is adopting the schema. Here, we will need to adopt one of those Darwin Core set of data tables. Here, we've chosen, again, the one that we use in EMODnet Biology, so the OBIS-env data format. So we would have an event table with only the fields that are relevant for the sampling events, to document the sampling events. We would remove everything that is redundant and not interesting here. We would do the same thing with the occurrence table. We would only keep the information that is related to the occurrence, but we would keep, of course, a field to connect to the event, so we know which occurrences happened where and when. And we would also have the extended measurements or facts table that can document the sampling methodology, the life states, the weight biomass, which we also changed before. And we would add those identifiers that help us, that come from BDC, from the NERC vocabulary server, to help us standardize everything that is in here in a somewhat longer table. This is a very brief example of what we can do with Darwin Core, or how datasets can be turned into Darwin Core. And I'm going to stop here for the moment and give the floor to my colleague Lynn to talk a little bit more about the DNA-derived data.
All right, thanks. So up until now, we've been mostly talking about samples and observations of actual organisms, but a lot of studies are dealing with genetic information instead. So information coming from, for example, metabarcoding, qPCR, or metagenomics. And while that information is usually, the raw information from that is stored in sequence databases, and it is used for phylogenetic research or molecular ecology, we could actually derive information from it that would make it usable in a more wider context, because we can derive occurrences from it. So we've already seen what the occurrences are. So basically coordinates, dates, and taxonomic information. So we can derive that from sequences. And if we submit those derived occurrences to a biodiversity database, we really make that information useful to a wider range of researchers, because not everyone is going to a sequence database and is going to perform a full bioinformatic analysis to get that information. So if we make it available in a biodiversity database, that's a very valuable thing to do, especially for undescribed and inconspicuous taxa that are otherwise hard to observe. So it's very good to submit that information to a biodiversity database like EMODnet Biology. So to submit it to a biodiversity database, we will need to transform to Darwin Core. So we've already seen the OBIS-env format. So there you have an event table, an occurrence table, and an EMOF table. So but since DNA data has a bit slightly different information that is needed, you will need an additional extension, and that is the DNA-derived data extension. So that extension contains terms from a few different standards. It contains terms from mixes, so minimum information about any sequence, from GGBN and from MIKE. So you might be familiar with some of those terms if you're used to working with DNA data. You can also click here on this list, and it brings you to the extension, and it will show you the full list of terms. So there's actually quite a lot of terms in there, I think more than 200. So those should be able to help you properly document anything you want about your DNA data set. This extension, it's an extension, so we need to plug it, connect it to a core. And initially it was meant to work only with occurrence core, but there has been a recent development that now also makes it possible to connect to an event core. But that's very recent, so we are still working on updating the guidelines and providing examples. So stay tuned for that. Then how do we actually perform this transformation to Darwin core? It will be mostly very similar to what Ruben has just showed you. I think the main difference will be that your initial data will look different. I think most of the time the data will be spread in a lot of different files. So I put an example here on the slide, and you see the data is spread across 4 different files in this case. So we have an ASV table, we have a sample table, a taxonomy table, and a FASTA file. So that information is in all these different files. So they actually need to be connected in a way, and they are connected by identifiers of the ASVs and the identifiers of the samples. So if you use those identifiers, you can connect all those files together again and bring the information together. So where are occurrences? Like in blue, I highlighted an example. So here we have 51 reads of ASV number 5. So that's actually a taxon day with this sequence, which is found in sample 2. So that's found in site 2 at this date, this time. So we have a taxon, a date, a time. So we have an occurrence. We just need to connect the tables. So to do the transformation, we combine the tables. Also, you notice in the number of reads, you will have a lot of zeros. Those can also be removed. Otherwise, you will get a lot of a very large data set. And then the steps are basically the same as Ruben has explained. You map the fields to Darwin core terms. You get the correct structure. Here, for example, an occurrence core with a DNA extension, and you standardize the content of the fields. Then also another step is enhancing that data set, so adding additional fields to the data set. So that's the same thing as Ruben has already showed. But in the case of DNA data, there will be a bit different requirements on which fields you should add. There's a lot of other information that is important to add, like, for example, the link to the raw sequences, the primers, the target locus, the total number of reads, and the bioinformatic methods, for example. So to know which information you need to record and to which Darwin core term you need to map it, there's actually a very good guide that has been published by GBIF in collaboration with OBIS. So if you click on the link, you can find that guide. And that guide really contains all of the details that you need to transform your DNA dataset to a Darwin core format. So here's another slide with the resources for the DNA-derived data. So the main thing is the guide by GBIF, but the other ones you can have a look at those as well as if you're interested. Something important to note on this is that the inclusion of DNA-derived data in biodiversity databases is quite a recent development. So a lot of the guidelines are still evolving. So if you have any feedback on the guidelines, it's always welcome because they can still be improved. So that was it for the DNA data.
Thanks a lot. Quite nice. I'm going to continue with the rest of the presentation, and this one's going to be, I hope, a little bit shorter and dynamic, since we have a couple of demos here. Before the demos, I will just give a small theoretical part about publishing data and about the quality control, and then we can see how much time we have. And since we started a little bit late, so we will see how we can manage it. So regarding publishing data, I would like to talk about the Integrated Publishing Toolkit, or IPT, tool developed by GBIF, and the definition here is a free open source software used to create and manage distributed data repositories that share biodiversity data into the GBIF network. So that's what we could do with IPT. Now, in short, it's a data repository that helps us to create Darwin core archive files. It also helps us to create data paper templates. So this can be very interesting for a number of you. And it allows for an automated flow to GBIF and OBIS. Now, of course, IPT is limited to the GBIF formats formats of Darwin core and the GBIF scope. However, this is very wide, so it's going to be useful for many of you. And I think recently they've integrated not only Darwin core but also frictionless data. I think so. So it's also evolving. This community, the GBIF community, is very active, and they work collaboratively with the TEDWIC community, which are the ones that maintain Darwin core. So they can also be requested a lot of features and support. I hope Niels agrees with me. Now, how does it work? It has 4 simple steps, or I've simplified it here. It has a lot of more other things that you can do, but this is a more simple way in which you can publish data. You upload your data, and this can be done in many different formats. Either you upload the files there that can be either text files or Darwin core archives themselves, or you can set up a direct database connection, right? Or you can add a URL that contains the data frames of your data tables. Then you will need to map these data tables to Darwin core. This is something that can be done automated if you already had your field names in Darwin, like using the list of Darwin core names, or you can do it manually if you want. And it also allows for data modifications afterwards, or you can do actually this one before. You would have to fill in the metadata, and this can be done manually. They're going to have a few text boxes where they're going to ask you what's the title of your datasets, what's the citation of your datasets, and you can input them there. Or if you've already created an EML file in a different way, you can also just upload it, and it will interpret and understand it. And the last part is just clicking the button of publish. And this one will also run a number of quality checks, mainly on integrity, dataset entity integrity of your data tables and metadata. So as you can see, it's very simple. Later on, I will show you the platform. But first, I'm going to finish with the last section of the presentation, which is quality control. And quality control in data management is a process of identifying and correcting inaccuracies or inconsistencies in data to ensure its accuracy, completeness, and reliability. Now, the rationale for this is, I have a couple of examples. The one in the left, you have a data set that comes from a monitoring in Svalbard, in the Kongsfjorden area. However, we found in this data set some point in Southeast India. Now, I imagine you can guess, but it's a very simple and common mistakes. And the latitude and the longitude have been switched for these number of records, creating erroneous information. And the case in the image on the right side of the slide, you see a monitoring that was supposed to happen in the Belgian part of the North Sea, so here. And however, there are some points in Great Britain Island. So the reason for this also very common if people don't pay attention, which can happen when you deal with large data sets or not with standardized or proper methods. It's that the plus and minus signs were switched in the longitude. A third example show us the difference in number of records before and after quality control. So here on the left side, we have the before before the quality control was applied, and here the actual number of organisms that were found. As you can see, there is a decrease. And such taxonomic quality control is highly needed since, as in this case, a misspelled or obsolete name could be compared to the introduction of a rare species, which can have adverse effects on biodiversity calculations, as you see here. Now, on the question, how do I start? What does it need to be QC'd? Well, everything, sadly, everything. However, there are a number of guidelines that we can follow, right? So if we have a standard, we just need to follow the standard. And some of our fields in our standard are going to have specific format. For example, Darwin core and has a requirement where the latitude and the longitude needs to be in decimal notation, the dates need to be in ISO 8601 format. So the standard itself already gives us some guidelines on what can be checked, right? Also, unique value fields, for example, the identifier fields need to be unique. We need to make sure that they are unique. We need to make sure that there are not duplicated records or redundant information. We're going also to check if there are impossible values, for example, dates in the future, very common if you use the auto-increment function of Excel, or outliers, mandatory data that is missing, or data that would make your dataset more complete or without which it would not be properly understood, such as the units of a measurement. And now, for this, we in EMODnet Biology have created a tool. It's called the BioCheck tool. And it uses as input a zip file, which is going to be a Darwin core archive file. However, it's based on an R package. So you can also, you can either have it as a web application, or you can read it in R and run it from R if you use it. Now, the BioCheck tool provides first an overview of your datasets, and second, a list of issues. Those issues are going to detect inconsistencies of inaccuracies in the format and integrity of your dataset, but also in the taxonomy, the biogeography, or those parameters that you have added. So now this is the end of the presentation, but I would like to have some little time for the demos to make it a little bit more visual. I know, I imagine, Marina, that we're a little bit late, but I also know that we have a half an hour coffee break later on. So do you mind if I continue for 10 more minutes, maybe?
Yes, sure, please. Go ahead.
All right, and apologies for extending a little bit on the time. I think now I'm going to stop sharing my presentation here because I'm going to start sharing. Let's see. Because I'm going to start sharing my browser. Give me 1 second, and I should have it here. So could you confirm that you can see the IPT page?
Yes.
All right, awesome. So well, this is how the IPT to create a new IPT resource or dataset. That's the name that the IPT gives to datasets. Quite simple. You give it a name and you select which core you want to have. So if it's going to be an occurrence core, a checklist, a sampling event, metadata only. And then if you already have a Darwin core archive file for that datasets because you created on your own, you can upload it here. If you do not have it, you can just click on create, and it will take you to something like, come on. Okay, I'm going to quickly log in. Sorry about that. I was logged out while I was presenting. So we have here, if we go to manage resources, let's see, this one. This one is the one I just created before the presentation to show it. And as I said before, those 4 steps that IPT has to create a Darwin core archive file or to publish data are these ones. The first one, source data. Here you're going to add your files from your computer. You can choose the files here. It's going to take you to your browser, and it's gonna ask you to select some of it. Then it's gonna put them here. You can watch them. You can explore them. I'm not going to show everything here because I don't want to lose too much time. But this is basically the files that I had in my computer uploaded. After we have the files here, we can map them to Darwin core. And here we see, we can have a quick look at it. This is how they look like. They have an ID, they have an institution code, so all Darwin core fields. And they also allow to change the mappings if you need to. So for example, the institution code of Darwin core has been mapped to a column in my dataset that is also called institution code. This has been done automatically, and I just have to click and save and back. We also have to upload the metadata. So for example, here I have already uploaded it, but the way it looks like is just here text boxes, as I said before. Here you write the title, write the license, you put in the description of your data set, and you have a lot more information: geographic coverage, taxonomic coverage, keywords, associated parties, project data, sampling methodology, citations, etc. Here in this format, it's going to give you the option of inputting all that metadata for you to make your datasets understood. You can make your datasets public or private, and finally you can publish it. Now, if your dataset is marine, you can also add the OBIS network to it, and then it will automatically be also sent to OBIS. And that would be all. I mean, you can just register here. Once you register, it does. Once you register it, it also sends it to GBIF. So you would need to do this one. I cannot do it here because this is test mode IPT. But you should be able to do this if you have the information about a GBIF publisher that you're using, for example. That'd be a very simple way of talking of showing how the IPT works. Now, let's see if I can. Well, yeah. Now I'd like to show the quality control tool or BioCheck tool. As you can see, it's a web application. If we go to the About tab, it's going to tell us that this comes from an R package that you can also use. It also has tutorials on how to use it and how to interpret the data and how to read the error messages. So all these error messages are going to be can pop up in your datasets if you have not done something correctly. And the input for this one, the input for the BioCheck tool, for this quality control tool, is either the URL of the IPT resource, so the one that is generated by the IPT, or you can also upload the file, a Darwin core archive file, if you want. Now, you click and load. It takes a few seconds, and that's going to depend on the size of your dataset. And once it reads it, it's going to tell you some information about it. The first tab is going to be a data overview, and the rest of them are going to be error message. Well, some information about the errors or potential errors that you can have in your dataset. So it's going to tell us that this dataset with this name has one cruise with 27 samples in it. These 27 samples are material samples, and 404 are present, and 3 of them, we don't know what they are. So this would be so they are NA. So this would be the first thing we can check. It's also going to provide us a list of the measurements and facts linked to our events and EMOs and occurrences. For example, here it provides the gear, and it tells us if it's properly mapped to the BODC vocabularies. So, for example, if the gear is equivalent to a sampling instrument name, then we're fine. This happens for 27 records. For the information that has that is numeric, it also provides a minimum and a maximum value, and also the units, which can also be compared with the units of that come from BODC, from the parameter that we've chosen. So if they are equivalent, knots and meters per second, then we're fine. It also contains this, for example, live stage stakes, everything that has been. Here we can also notice if something is a little bit weird, like abundance per something. What is this? Of course, this is a mock data set, not a real one. It's also going to provide information about the values, not only the parameters and the units of the EMOF table, but also the values. So it's going to tell us that we have added as a value for sex and F. This could be a female, and it's confirmed by the control vocabulary that has been chosen from BODC NERC vocabulary server. It also provides a map that is going to tell us if our records fall in the ocean or not. And the thing is that EMODnet Biology, of course, works with marine data. So if we find some information on land, this is likely to be an error for a marine dataset. Although, of course, you can test any dataset here. You can just ignore this issue. Well, this is a temporal cover of the dataset, but because this dataset only has 1 year, it looks like a box, but it should be a bar plot. And it also tells us here what the metadata coverage of our dataset is. So if they are equivalent, that means that we're fine. And it also looks at it also looks at the taxonomic cover of the dataset. So if you have a dataset about cetaceans, but you find polychaetes, you know that something is weird about that specific record. And the next tab, and I'm going to stop in this one because all the others just develop on this one. It tells you a number of issues that can be found. So, for example, duplicate EMOFs, record linked to an event, or there is no sampling instrument name, Or, for example, the biological quantification of 0 has been added for occurrence status that is present. What this means is that you've added an occurrence as a presence. However, you say that its weight, for example, or its length is 0. If something is present, it's probably going to be repeated something very small, like 0.0000 something grams, but it's likely not going to be 0. So this is one of the common errors when people just round up values. Coordinates are located on lands, event dates is not a valid date, for example, it doesn't conform with the standard, etc. So this tool itself is already going to give you a lot of information on your dataset, which is very useful if you deal with datasets that have several hundreds or thousands of records, and it's difficult to search them visually. So this one's is going to help a lot with that. And of course, it tells you which are the records. It's going to tell you the event ID or the occurrence ID have these issues, right? So you can filter here by what you're interested. Yeah. So maybe I think that would be all. Maybe if you can also click, I haven't mentioned it, but you can also click here in event hierarchy 3 equal yes. And what this is going to provide, it takes a little bit longer, but it's going to provide structure of our data sets. For example, tell us that we have an event type, an event here that is a cruise, inside which we have samples which have abundances, observed individual counts, wet biomass information, etc. So it helps us to have a clear way of how our occurrences and events have been linked to each other. And with this, I'm going to stop. Thank you very much for listening. And if you have some questions, I'll be happy to answer them.
Yes, please, Angel.
Yes, I have a question because I understand that if we submit data to EMODnet, these data are shared with EuroBIS. It's not necessary to send again the same data to EuroBIS. I'm right?
Absolutely. Yes, you're right. So EMODnet Biology uses the EuroBIS infrastructure. So although you send your data to the EMODnet Biology mailing list, we're going to store it in the EuroBIS data system. So it's going to also be there. But since it is also in the EuroBIS data system, from there it's going to flow to OBIS and GBIF. Or we can actually do that from IPT. So if you send your data sets to EMODnet Biology, you will end up in EMODnet Biology, but also in OBIS and also in GBIF.
And my second question is there any relationship with the EMBON from Euromarine or something like that? Is EMBON?
So I was last year providing a similar type of workshop to the people from EMBON, right?
Mm-hmm. Yeah.
So and a number of them, I think I've seen Nestle here somewhere. So some of them would be submitting data sets to EMODnet Biology as well. So we do have some links with them as well.
Good, thank you.
Cheers. Niels?
Yeah, so thanks for a great presentation on the IPT and tools. So we have been piloting the meta barcoding data toolkit, and this is getting in a quite advanced state. So anybody who is working with eDNA data might be using that toolkit, which facilitates the work quite a lot. From there, you can also export Darwin core archives and then supplement the archives with the probably with the OBIS environmental data, so that you can enrich the output from the metadata, meta barcoding data toolkit with the environmental data specific for EMODnet.
Yeah, great remark. Sophie?
Yes, hello, good morning, everybody. And thank you, Ruben, again for a very clear presentation. I have a practical question from a scientist like myself that is generating eDNA data sets from the marine environment. We have recently uploaded our data to GBIF, but I understand now that we have GBIF, OBIS, and EMODnet Biology. Is there any guideline on where we should actually submit our eDNA data, or am I free to choose where that I submit?
I mean, the data is going to end up everywhere. Now, I know that ILVO has a contract with EMODnet Biology. However, it does, I would say, please, Joanna, correct me if I'm wrong, but the data is going to end up everywhere. What you've done is you've submitted your data, you've put your data set in an IPT, and from there it has flown to GBIF. Now, what's going to happen if you have added the OBIS data network to your IPT data sets? OBIS they're going to receive this data set as well. They're going to send us to the EMODnet Biology team a message about it, and we will also ingest it in EMODnet Biology. So that's also a channel that it can follow. So the things that we're all in contact with each other through a number of methods.
So it doesn't matter, or do you say, yeah, you are actually making a detour now, and it's better just to immediately upload it to EMODnet Biology?
I'm going to stress that if you have again a contract with EMODnet Biology, the datasets need to be formatted according to the EMODnet Biology guidelines, which can be slightly different sometimes to the ones from GBIF. As I've talked about before, we follow the OBIS-env data format that can be complemented with the eDNA table. So I would say maybe for EVO, and depending on which data sets we're talking about now, we would need to, even if you submit it to GBIF, they would need to have the format that complies with EMODnet Biology. Do you want to add something, Joanna, about it?
No, that was exactly it. I wanted to add that for this project data to be available in EMODnet Biology, you have to ensure that it complies with our criteria, the quality criteria especially. So it doesn't really matter. Yeah, it doesn't really matter in which IPT it is, or where you submit it first, but it does need to comply. Yeah.
Yeah. Okay. Thank you.
Luke.
So but the data, if you publish them in GBIF, are they only moving across to OBIS if you tag them with the OBIS network, right?
Correct. The thing is that is the way we have to realize that the dataset that is marine and it belongs in OBIS and EMODnet Biology is in GBIF. So as you know, GBIF has a lot of datasets, and this is a very easy way for us to filter the ones that we need in EMODnet Biology and OBIS. So we are encouraging all the marine data users that publish the datasets through GBIF to also use the OBIS network when they are publishing in GBIF.
But then if they forget or don't, I just wonder why we use this network and not use some polygon that would just grab everything from marine coordinates. Obviously, you have to have something else as well, because a polar bear could go on land and then it's still a marine resource. But
absolutely. We also have a number of tools. For example, as you say, like a polygon that has the European waters. We do spatial geom with what is interesting for us. And however, that takes a little bit more resources. So we're trying the community to contribute, right, and to know where they need their data to be sent to. But indeed, we also have our own way to filter GBIF data to find European marine data sets.
Oh, good.
Cheers. Meike?
Yes, Ruben, thanks very much for a very, very good talk. I have also 3 practical questions, being the person trying to get people to comply with these standards. So the first is, oftentimes the data submitted to us is added value data. So it's, for instance, downloading all abundances from OBIS and then converting them to carbon biomass. But then we often run into trouble that we can't put the data back, because it's not an original observation. We're not the PI on the original data point, but we've done something to the data. Is there any way to track provenance, having a QR code for data reuse that you foresee for the near future?
That's a really good question, and I'm not really sure I'm the most indicated person to answer. But of course, in the metadata of our datasets, we do have links between different IPT resources or datasets. So that would be a way in which we can keep track of what has been done. Yesterday, I showed when I talked about the Integrated Marine Information System, or EMIS, that we have here in the Flanders Marine Institute, that they do keep like different versions of a data set. And there you can also have in another formatted, a data set, the same data set formatted in a different style or with other things. although yeah, I'm not really sure how that can be done without issues. That is one of the problems that we have, keeping provenance in our data sets. Also, we work with a lot of data aggregators. We work with GBIF, we work with other OBIS nodes, we work with ICES, for example. We work with people that, as you, just aggregate data from literature. And so it's also very relevant to know that we are not, to make sure that we're not republishing things that are already there, maybe creating duplicates that are masked because we've changed slightly the format and we have not kept the identifiers from the beginning. So this is I would say this is a big problem that we all have to tackle together.
Okay, second question is that people are now generating data at an exponential rate, and oftentimes the bottleneck is scientific publishing. How do we deal with this fact that oftentimes we would have a lot more data if it were also to include unpublished data?
And for the, well, I mean, as long as we have everyone using the tools that we've suggested, like the integrated publishing toolkit, so IPT, it's a matter of, so the individual researchers submitting their data sets, the data sets will find their way to OBIS and EMODnet Biology if you put the, and GBIF, of course, if you put them in an IPT. So there you will not have that much weight on the data managers at this side. However, something that I've been thinking about for some time is about the quality control, because GBIF, OBIS, and EMODnet Biology have some quality control, but some of them allow the publication of data sets with known errors. They just highlight them or they add quality flags to them. And however, I'm also interested in the prosecution of, I mean, of the researchers or the data managers that have submitted those data sets. So those issues are not only flagged, but can also be fixed.
But is there a flag or a column to say this is an output of a regular monitoring program that has not been through peer review, and that other data has actually gone through peer review and has been quality checked by the scientific community?
You do have some fields in Darwin Core that, or in the metadata, you can also add yourself to specify that the data comes from, for example, citizen science projects that don't necessarily always have a validation, or with the recent how's it called, the taxonomic identification that is performed by algorithms. Sometimes they need a validation by a human, and Darwin Core recently added some fields also to specify if that record has been validated or not. I think it's called identification verification status or something like that. Those fields will help us to be a little bit more sure about the taxonomy that we are using, or for the users to be sure about it. But it's a matter of documenting everything, or we do not always have fields to put all that information. However, everyone can get involved in the TEDWeek community, which builds upon the Darwin Core standard. And there are a lot of conversations happening in GitHub about all these topics, how to document the provenance, how to document the issues that you've just been raising.
And last question. So I totally love the IPT and the quality check tool, but I feel that I don't get my data contributors to use it because it requires a lot of knowledge already about data.
Well,
is there,
I'm sure, oh, sorry.
Sorry, is there any way that this could be automatized with a sort of language-based AI that would sit together with you, open a window, say, hi, I'm your assistant. Let's have a look at your data.
Yeah.
Because I feel that I don't get people to do the curation.
Ruben, I'm sorry. Sorry to interrupt. Can we, Marina, can we just move to coffee, because this is extending a bit, and we'll get back to your question, Maker, afterwards.
I have a very simple answer.
Okay, yeah.
Yes, but there are also plans, EMODnet, at a wider level at EMODnet, so I don't want to say much at the moment.
Yeah, okay, I like that look. So chat IPT to answer Make, apart from all the documentation and guidelines that we have, would help you. However, it's still in very early stages. So, but it would help you with some AI to format your data sets. That would be all. And look, I hope we can talk later, but I agree with Joanna. And we're already a little bit late for the coffee break and whatever, and the rest of the presentations. So if you want, send me an email or something and we can discuss. Thanks a lot, Marina.
Okay,
Thanks
okay.
a lot, everyone,
So, but I
questions.
propose that we reduce the coffee break because we are a little bit in a hurry. So let's do 15 minutes. We come back at 11:45. Okay. So we anticipate a little bit with the Salva presentation. Yeah. Thank you. So in 15 minutes then. Cheers. Cheers. Okay. Okay, I hope you are all back. We were a little bit ahead of behind schedule, so now we will start again with the presentation by Salvador Fernandez from Blitz on the biology products and guidances. Please, Salvador.
Hi. So I moved rooms and now I'm with Ruben and Lynn. Okay, how do I share here? Yeah. Yes. Okay.
Wait a minute, they don't see it yet.
Yeah, we need to share. I hope you rested during this break.
Yeah, there was enough information before.
The one, this
slideshow.
This
Yeah.
one, oh yeah. All right. All right. Can you see our screen?
Yes, now we can. Yes.
Yes. Okay. Thank you. So yeah, up until now, my colleagues have been talking about how data is managed in EMODnet biology, how it should be formatted, all the standards that are followed. But what I want to talk in this short presentation is what happens after. When you have collected your data, then you, in a typical scientific analysis, you transform this data, you change things on it. For example, you can make a model, you can make some kind of statistics and get some data out of that, right? And then you typically get to a file that has all this information, all this analysis, and this is what we normally use as a EMODnet biology product. So, for instance, okay. Sorry. So yeah, a typical case that we find the most is this: we get to a file, a special file that contains information about something. For example, this example that I put here is an external product, external because it's not a direct partner of EMODnet biology, so we didn't create it within the project. It's someone that's outside that did it, but we are really happy to collaborate and ingest it. In this case, these are habitat suitability maps for 30 different species and at different levels of them. So yeah, just this, if you are familiar with your spatial data, this will be a raster refreshing. You have this is an image in which each pixel contains some information, and each pixel is put somewhere in the world, okay? So it has a location assigned to it. So in EMODnet biology, when we have raster data, to publish it, we need to put it in NetCDF format. And I will basically talk all this presentation about NetCDF. There are other cases before that in which, for some reason, the output of your research can be in vector format. We can also ingest that. And also, it's interesting to mention that we also provide, as data products, is not, in a sense, a data product, but it's software. And we create some R packages, some Shiny applications, but it's not limited to R, it's just because the language more knowledge during the team that people know the most. For example, here we have the EMODnet WFS is a package to retrieve part of data, vector data of EMODnet, but also we have a package to work with trait information, sometimes series in an R Shiny, and we have a few of them. But mostly what we're interested in NetCDF files that contain models, contain statistics, contain the result of biodiversity studies, these kind of things. So yeah, I will talk how it should be formatted. How, if you have one of these results of your analysis and you want to publish it in EMODnet biology, you should put it in NetCDF. But we use if you don't know much about NetCDF, you can select different structures for data in NetCDF. NetCDF is just a file format that allows you to store multidimensional data. So if you remember what we were talking before about the Darwin core, you can have your event date, so you have time, that will be a dimension. You can have decimal latitude and longitude, these are 2 dimensions that put you in space, where is your data in space. And you can have other dimensions like time and depth. You can have, and what is more important for our case when in biology that I explain later is that we consider the taxon to be a species, the taxon to be a dimension. Because what we're trying to do is like we put all the data in one single NetCDF file instead of separating in different files, okay? Refreshing about NetCDF, this will be the typical output that you see of a NetCDF file if you read it with most of the softwares, can be R, Python, or specific tools. It looks a bit what you see. You see dimensions that, as I mentioned, they define how is the shape of the data. And here you put measurements like location, time, but it can be many of them that are not that common, like can be species, as I was mentioning, or climate change scenarios even. Then you can have variables in which you put your actual data. The example that we put here is temperature. You will see that here you can put also habitat suitability. You can put, I don't know, metrics like Shannon index. You can put abundance, different things like that. And a special case, because I will mention several times, is coordinate variables. This is just a variable in which you put the data for dimensions. For example, if you create, in this case, an X dimension, it's not longitude, but okay, it's the same, it's fine. So you create an X dimension, but you also need to create an X variable. This is a one-dimensional array or variable in which you put the data for this. So you will put in this case, for example, the longitude of it, the time of it. This is a requirement for NetCDF files. Now, what did we choose NetCDF files? So as you probably are aware, at some point in EMODnet, it was decided that all the portals will move into a single centralized portal. Most of the servers and web services, they are still maintained for all the thematic logs. But in between, it was decided that EMODnet will create an ERDAP server. ERDAP is just a server that allows you to put both NetCDF files, but also tabular data, can be tables. But for our case, it's really important that it allows NetCDF. The good part of it is that you put it in the server and then gives you both a website in which you can like just search for them, do your thing. But it also gives you an API, which means that you can read it through programmatically through programming languages or remotely in any other software. You can like even use it in your own website, right? That's what happens actually in EMODnet. And also what is nice about ERDAP is that it allows you to do subsets of the data. So once it's there, it allows you to say, okay, I want only this slice of latitude, longitude in this place at this time. And then you retrieve only that. Like it allows you to retrieve only small subsets for your case, and you don't need to retrieve the whole file. That normally tends to be huge. Normally it's like gigabytes. And so what we decided in biology is that we will follow this structure of NetCDF, because it's also what chemistry and physics were doing. And we thought that ERDAP will solve most of the issues that we were having, and it will be the best way of serving the data. But also, yeah, this is maybe more relatable. This is what happens often. What you see on the left side is just like a folder with a lot of data. This is a real case that I got this year for a standard product, in which like every folder has is named after the species. And then inside every folder, you see, as you can see, like many files. These are AZTI files. These are like, it's a SEMOF-TIFF. It's a geospatial raster. So inside of those files, there are latitude and longitude dimensions. But then the other dimensions, which in this case will be, for example, time, you can see there 2020, 2030. That will be the time. And that's something that you know because you know the project, because you have read the paper, because or someone has told you. Otherwise, there's not the metadata that tells you anything about this. Also, what you can see some letters there, A1B, RCT 4.5, etc. Those are climate change scenarios. Again, that's something that you know because you have read the paper, nobody told you. And also, when it says training, because I have read the paper, I know the training is the data that was used to fit the model. So, and it was data like from a random period of time. Random in the sense that it's the data that they could find or they collected, okay? While the other ones are more like fair-related, okay? Because are like climate change scenarios for the future. So yeah, this presents a few problems. That feels like there is no standard way of organizing these files. In this case, it was exactly like this, but in other projects, probably it's exactly in another way with different file name conventions. So it's really difficult to work with this unless you have the knowledge of the project, because also there is no metadata assigned to it, and also the dimensions that I'm talking about conceptually, because they're using AZTI files, it only allows longitude, latitude, and longitude. So you have two-dimensional files, and the dimensions are not contained in a single file, so it makes it more difficult to work with this data. So from this, we move to an NetCDF structure, in which, all the data is in one single file. And in this case, because there were conceptually 5 dimensions, and I will say latitude and longitude, those are typical time that we have seen. Time meaning the time for the train, meaning they use, I think, from 2000 to 2016, something like that. But then also prediction for the future, for 2020, 2030, 2050, etc., etc. But they also have 49 different species, so that's also a dimension. And on top of that, they also consider 3 different climate change scenarios or emission scenarios. So that makes a 5-dimensional array for this data. Once you put it in this way, it's much easier for ERDA, for all the people that know this format to work with it, because also it allows you to add metadata. So as you can see, for example, the latitude, it has units in the Greece north. It has a standard name. Now I will explain. It comes from the climate forecast convention, and different things. The good part is that you can add also metadata to this. Also, it's not included here, but you can include global attributes. So it would be like, as Ruben was mentioning, you can include inside the file, you can include the title, the citation, the license, and actually anything that you will want to include. And to be understandable for machines, for people, for everyone, to be interoperable, of course, it needs to follow conventions. In this case, it's a bit more complicated than the tabular data because there are no conventions for biological data. There is no well-developed conventions in NetCDF and in multidimensional data because historically this came from oceanography. But still, you need to follow these conventions, and it's really important because it's a technical requirement. Otherwise, it will not work. We cannot upload it and we cannot put it on a website if it doesn't work, if it's not following these conventions. And these are things that we are also learning along the way, since that we try and error and we learn all of this. Okay. So there are 2 main conventions that are important. The first one is COARDS. This started in the 90s. Actually, I was a little kid, and it's the most basic set of rules for organizing spatial data. So it describes what I said now. For example, the coordinate variables, like how the basic ones are longitude, latitude, depth, and time. And it's important in this convention that these dimensions, these coordinate variables that define the dimensions, they must be numeric. This is important, remember it, because it will have some effect later in our work. Other things that are interesting about COARDS, well, the numbers are stored, the time are stored as numbers, so you cannot put a date in the ISO that EMODnet Biology normally follows. You need to use this format. That's, for example, days since a specific date, but there are tools that convert dates to this, so it's actually fine. Okay, and also all coordinate variables require units, even if you don't have, in the sense that, for example, if you are talking about taxon, there's no unit for taxon. Taxon is 1 or 2 or 3, but there's no for a percentage. While, for example, you're working with let's say, with depth, of course, the units can be meters, can be anything like that. So that's for the COARDS convention. But then more fast forward to the future, there is a more recent convention that is more developed. It's a climate forecast convention. This builds on COARDS, but it's aimed to increase interoperability. So this includes things like a table with the standard names for different variables. For example, for temperature, there is, for many things there. But you will see how for biology, we don't have all of those standards. And that's something that we are working on. And what is interesting and they affect our work is that coordinates variables, in this case, they can be characters. They are allowed. Other things, yeah, like it provides a different ways of, like, for example, adding global attributes to explain the data and how to handle missed values. So yeah, in a more practical case, I will show why it was important the difference between the coordinated variables in character. This is how the CF convention defines how to add biological data to a NetCDF file. Bear with me. This is a base example. So what you have in the dimensions is only a time series, so we only consider time. You can ignore the string dimension and just focus on the taxon. So basically, we have two different species here. To define these two different species, you need to create two variables that are the taxon name and taxon LSID that Ruben has mentioned before. So it's the identifier, in this case of worms, and the taxon name, just the name of the species, right? What is the problem? That it doesn't follow the COARDS convention because the coordinate variable taxon, its characters. You will just need to use taxon name or taxon LSD, and both of them are text. So when we upload to our systems, we found that it fails because it doesn't follow the COARDS convention. So what was our workaround for this? We made the taxon dimension to be numeric. Okay. What was our way? Instead of taxon, we call it FEID. And then in FEID, we added the FEID. So just the number that identifies the species in worms. We also keep the taxon name and the taxon LSD, so we are following the standard of CF. This way we can make it work when upload it, and also because we have the FEID, we can use it to retrieve data dynamically from worms. More challenges that we found is, as I was mentioning, for the time dimension, many times we have projection for the future, and then like what they consider, for example, a decade. They consider the problem with the time dimension in NetCDF is that you need to add it as a single date. You need to as a day. Even you can go to minutes, right? But of course, now we're not talking about a date in particular, but we're talking about a decade. So luckily, the CF convention gives a way of how to structure this. That is through climatology, they call it. So what you do is you add your time dimension. In the time dimension, you add the midpoint of the decade. For example, in this case, the first one is the 2045. And then what you do is you create another variable with two dimensions. In this case, it will be time, and also one that I don't know why, but it's called NV. But just in this case, it's just because it has the two extremes, right? It's at the beginning of the decade and the end of the decade. So you need to have two dimensions there. And then you call it you use it to a variable called climatology bounds, in which you add exactly the bounds, the beginning and the end of the decade. All right. You add an attribute in your data in which the cell methods attribute in which you specify how this was computed. This is an example. And in your dimension, you add the climatology attribute in which you point to the variable that contains the bounds. In this case, like this, right? But we found even more cases. For example, I was saying the climate change scenarios, like conceptually, this is a dimension, but there is no standard for it. And this happens many times that the data that is produced in an analysis is model data that makes different projections for the future for different climate change scenarios. And we found even other cases that I'm not going to go in detail, but it happens often. And we still need to have one single file. We still go for this approach. Our solution so far, it's just to create a dimension with the name that we prefer. In this case, we use emission scenario. We add a coordinate variable that is just numbers, just like 1, 2, 3, 4, 5, because remember, because of the COARDS convention, it needs to be numeric, otherwise it doesn't work. So we add this. We add a description attribute in which explain, okay, 1 means this climate change scenario, 2 means this other climate change scenario. We also had another variable. This is our approach. This is not really necessary, but it's our approach in which we just add the explanation of every emission scenario in a longer text. And that's it. That's our solution. Now, this is one of the limitations that we're finding. Other limitations that we're finding is that, as I was mentioning, the climate forecast convention offers standard names for the different variables, but there are no standard names at the moment for common things that are done in biodiversity and biology. For example, there's no standard name for habitat suitability or probability of occurrence that comes out of a habitat suitability model. There's no Shannon index. There's no species loss. There's no many things, right? Yeah, this is a problem, and this is something that we're going to be working on in the next years, especially through the editor project, in which like they are requiring us to adjust to standard names, and we believe that we're going to do this. We're going to come up with different names, and we're going to contact the CF community, and we're going to propose this. We're going to try to align with Biodiversity, but that's a story for the future. Now, there are many issues with this, of course, and you may think, like, why are we still trying to use NetCDF? We are not using another format. Personally, I still think that NetCDF is the right way to go because it allows many different things that you cannot do with tabular data, and also it's much more compact. It allows you to have much, large volumes of data in these files, and it's easier to access them in smaller chunks, right? So what I think here, what we do is it's being pioneer in this use of NetCDF for biology. So yeah, instead of being sad and feel daunting that this should not be the way to go, I think we should still go for it. And but keep in mind that we are learning along the way. There's not everything has been invented. So yeah, like if you want to contribute to EMODnet Biology with your own data products, that I will be really happy. But keep in mind that we need to contact, we need to talk to each other. You need to tell me what's your case, and we will try to find a solution within what we have until now. And we find a new problem, then we will try to find a new solution, right? So yeah, I added here some links. If we share the presentation later, have a look at them. This is our guidance up until now. We have a guide for NetCDF, how we need to construct it. I created a demo of how going from a table, a structure in that WinQuery format to a NetCDF file. So bear in mind that this is for products. This is not, if you are submitting data, then it goes through the way that Ruben explained. And yeah, we also have a template if you want to structure your project in a certain way. And voila. As I say, if you have any questions you want to contribute, the best is that you contact us, and then we review your case, and then we work on it, okay? Thanks.
Thank you very much, Salva. I see a comment by Lucas, if you want to say it. Yeah, please.
Okay, I don't really need to speak so much, but yeah, it's just really great to see that EMODnet are using NetCDF now. I'm quite heavily involved with the CF conventions, so yeah, let's talk in future.
That would be great. That would be really great because we need to contact we need to contact the CF because while I was talking about the standard names, that will be something that I think is really relevant, and we'll see in the future, definitely.
Cool. And if anyone else who's watching wants to learn more about CF NetCDF, I put some links to some tutorials I've made in the chat, so go and check those out, and you can find them on my YouTube channel as well.
Thank you, Luke. And there is a question by Angel. Yes.
Yes, it's a general question for EMODnet. I want to know how do you deal with the taxonomic issues? For example, in worms, sometimes they change the names of the species, accepted species, or sometimes they merge or they split species. How do you deal with this in the historical data sets you have, or in the data set we can upload there if there are some changes in the near future regarding this?
Maybe I can mention something about it. And when you're submitting, this is something that I didn't mention in my presentation today or yesterday because we didn't have enough time. But when you're submitting data to EMODnet Biology, the taxonomic information that you provide is not the currently accepted name of that taxon. The one you provide is the one that you have originally in your data set, and it has been identified by your taxonomist. Now, worms is going to change things and adapt. They do deprecate some terms if the community, for example, don't use them anymore or something like that. But the good thing of it is that they all link to one accepted name. So if you always point not at the accepted name when you're submitting the data, but at the original name, it doesn't matter what they do afterwards, you will always be able to point at the current accepted name whenever you look at it. That's why it's not recommended to use the accepted name for submitting data sets to EMODnet Biology. This is something you would use when you are working with the data, as in creating analysis or visualization tools afterwards for the other parts of the research. But if you always keep the original one, it doesn't matter if they, well, if they I don't know exactly how it works when they split it, but of course, this is a conversation that we can also have with the WORMS data management team and see what are their recommendations. But for us, just use the original one. And then if they change something, they should also document what changes they've done so we can track those changes to the current standard.
Yeah, thank you very much.
Cheers.
So you both gave very dense presentations that will be available both as recording and also as the presentations. So are there any other comments or questions? Otherwise, I proceed with the last one, which is just a sum up of what we have seen so far. Can you see my presentation? Because I cannot see it. Yeah, I hope so. Yeah. So over the years, EMODnet has provided a lot of guidelines and protocols, and so what has been presented by Ruben and by Salva before. So where are these tools available? Please check the EMODnet central portal. The information is available in two parts of the central portal, under themes, so directly under biology, or under data sharing tools and services. So in both systems. What is available? What are the key services available under the biology? As described before, the several web services, the IPT, the Integrated Publishing Toolkit, then the quality control tool that Ruben has explained, then the several web services, so WMS and WFS, the R packages, and the Darwin core file template. So all this information is accessible through the template. And now I would like to focus on some very important, very useful tools. So the online tool, which is the online course, which is available under the Ocean Teacher Global Academy, which I consider quite useful. Then our colleagues from EMBRC FAIR have already organized one first training, and the second one on FAIR data will be organized in April next year. And this is the link. And also the colleague from OBIS also have developed courses. So these are all available online. Then this is the templates or the Darwin core that Ruben mentioned. So they are available through the website with all the information, the description of the terms, what are mandatory, what are not mandatory, and so on. And then the guidelines for biodiversity data products. So what Salva has mentioned. And several tools. So all what you have seen in over these 2 days are available. And in this presentation, you find all the links. And also several tutorials. So on OBIS, for example, on the data format, on the vocabularies, for eDNA, on GBIF. So all this information is available. And also on the website, you can find informative material on the ongoing implementation of the process, of the project. So also the catalog of data that we provide, and also the news. Also, this workshop was on the news, and we will present a report after the workshop that we will receive, and I think it will be also available either as a report or attached to the news. And lastly, you have also seen in Johanna's presentation yesterday, where to find the data and the services. And this is an example of the map viewer with an information. And I think this is all, because this was just to recap what you have been presented these 2 days. And so all this information will be shared with you as soon as we prepare a small synthesis. So for the moment, I stop here. Now there is a lot of time for questions or comments. Joanna, you want to add something or to integrate?
Yes, we were just saying we look like a family here. But yes, I would like to just share a few remarks. So we are fully aware that this was maybe a bit overwhelming and quite dense in some points. Do note that the data training that we organize tends to happen over the course of 3 days. So what Ruben has done had to be condensed for a few hours and during the workshop mornings. So that's why it's so dense. Do check our resources that are available. One thing that we would also like to reinforce so that it's not forgotten and it's very clear. We do publish data that are collected in situ, new data collected. If you are working with data that are already published somewhere else, as we've mentioned yesterday, we need to assess whether those data exist in infrastructures like OBIS and GBIF, because we do not want to push duplicates into them. So the more information we have on the provenance of those data that you are working with, the better it is and it's easier to identify where they're coming from. Another thing that we had discussed as well, so it was the duplication. On the, I think we were discussing over break whether there was a bit of a confusion of what needs to be reformatted to Darwin Core. So the data, what we consider data is in situ observations or from samples that were collected in situ and are analyzed in the lab. Data that are created through the means of aggregated. Thanks, Marina. Through the means of through the means of aggregating data sets, published data sets, or models are not data. These are data products. We consider them as data products. They can still be published by us, but they do not need to be in Darwin Core formats. I think that was it.
Yeah.
Yeah.
I think that was it. And any questions? Yeah, we're happy to help as much as we can. Thanks, Marina.
Yes, well, we should underline again that this is the second workshop, but over the previous phases of EMODnet, we have organized several workshops dedicated to different stakeholders, and we need your feedback to understand if this was useful or not, if you expected something else or what you expect. So again, we will ask you to fill in a short survey, which will be sent to you afterwards, I mean in the afternoon, because we really need to tune our efforts towards being more efficient. And so we really appreciated this workshop because we are very glad we had a lot of interest, a lot of participants, but we have to understand if we were successful, I mean if it's useful for you, and how we can improve. I mean, EMODnet is going to be, there will be a new phase in the future, and we also have to design the new activities so we can tune some efforts to better respond to the needs. One other question, we will share the presentations by all the colleagues, by Ruben, by Salva, by Johanna, and so on. Are you also interested in accessing the pitch presentations? I mean, would it be interested for all of you to receive also the pitch presentations? This is the question to all. Yes, I see. Yeah, you can use the also the yeah the tool. Okay, so we will prepare a shared drive where we share also the pitches and the presentations. And I think there were also very interesting links and information in the chat, so we will also take that into account. Yes. And yes, can we also take a picture? Can we all turn on our camera and take a picture of the participants also of this session so that we can, yes. Thank you. Fai tu? Riesci a fare? Thank you. Thank you. Okay, I think we have completed all what we have intended to. So what you say, Johanna, we can say goodbye for today and start working on the minutes and on the report.
Yes, I think we can do that. Yeah, it won't happen. The report will not be published before the end of the year, just to let you all know. But we are aiming. No blue sky yet. It's yeah. We haven't decided on that yet.
No, but we can share. We can share a drive with the presentations and yeah.
Yeah. Well, yeah. I think we need to see what's better in terms of the least amount of work that we can do as well. But in January, I mean, it will be a news message, but at the very minimum, you will all receive an email to point you to the right locations, be it the drive or the reports and the recording as well. Yes, the recordings will be shared. Yes.
How is the question?
How? Oh, how? Sorry. I will need to. I will ask the EMOD if they can be added to the EMODnet official YouTube channel. If not, maybe through a drive, like Marina was saying, like a Google Drive or something like that. Yeah.
Okay, and I would like also to thank all the people who participated in organizing the questionnaire, the analysis of the questionnaire. Some people are not yet connected. My colleague Nicola and people from ILVO, and of course you all from Blitz. So thank you to all and all the participants who join us.
Thank you all. Yes.
Thank you. Much appreciated.
Okay, thank you very much.
Thank you.
Bye, everyone.
Bye.
Bye. Thank you.
Bye.
Thank you.
Bye.